So, good morning. So, as said, Karina, I'm going to try to explain all the challenges and the method that face the quality control of biopharmaceutical and all the methods that could be applied for this topic, for this purpose, sorry. So why we are in the industry quality control of biological or whatever the drug products are mandatory. They are mandatory to ensure safety and efficiency, especially in the case of biologics, such as a recombinant protein, as a monoclonal antibody, for instance. The active product is heterogeneous, structurally complex, and sometimes prone to degrade easily. So, to address this problem, orthogonal method has to be performed and orthogonal test has to be performed. So the main thing in terms of quality control is to ensure the purity of the compounds and also its identity. As well, different methods such as dosage has to be done. We have to also ensure that the drug administered to the patient is stable, not degraded and not degradable, and also to check his biological activity in the term of recombinant protein. At which step this process has to be performed? In process, so during, for instance, the upstream process, the development of the scale-up of the product of the drug, it has to be done after the purification process, after the formulated products, or also as well in the compounding product, when at the hospital, for instance, the compound is reproduced in an infusion bag, for instance. So, to go a little bit deeper after the previous slide, so as I said, quality control testing has to be performed to assess and ensure the safety and efficacy of the therapeutic proteins. It's also true that they have to check lot-to-lot consistency or batch-to-batch consistency. So the identity test that to ensure that the right protein is will be attributed to the right patient, for instance, to verify the biological activity, to ensure that there is no immunogenicity phenomenon, such as unwanted response after the administration of the drug. The purity has to be also checked to be sure that there is no contaminant, or there is also no, for instance, apparented drug, such as, I don't know, for a protein that could be a dimers, that could be a degraded form, a truncated form. So everything that could be and also every product that could be detrimental for the patient health. Of course, the dosage, to ensure that the right concentration of the purified protein in the final products, and to be sure that the right dose is administered to the patient or delivered to the patient. The stability also, as the protein, during, as we have saw yesterday, and maybe I will give you other example today, during the development, the protein is submitted to different stress. Whatever, during the development, during the purification process, as I presented yesterday, you know that, for instance, for the monoclonal antibody, they are captured once the harvest is collected. For purification purpose, the antibody is captured by chromatography with protein A, I think you remember, to capture selectively the antibodies, but then when you have to elute them, you must use acidic pH. And this acidic pH could be detrimental for the proteins. And you have also to relieve a potential virus activation, you need also a pH, acidic pH, that could be also detrimental for the protein. You have also to, so that's why the stability has to be, that is what an example, but you can also have a problem of stability once the drug is reconstituted at the hospital, because you have your infusion bag, this infusion bag has to be delivered to the patient. So you go through, the infusion bag will travel inside the hospital, and it could be agitated, it could be submitted to light stress, variation of temperature, so you have to be sure that your protein will not be, detrimental for the drug, for the proteins, for instance. So you have at many steps, the drug could be submitted to different stress. They have also to define optimal storage condition, and that could be this big issue, especially in terms of formulation. And at the end, the activity has to be checked to be sure that the drug, once it is produced, his affinity toward his biological target is constant. It's possible also to perform in vitro assay. Most of the time it's affinity test or sometimes test on bench on a culture stage, or in vivo assay, but it's a bit more difficult. So just to remember here, just remind you the overview of the all biological products that could be tested. So you have on the biological product on the left is all the natural products. That could be vaccine, that could be hormone or polyclonal antibody, that could be extract from plant or whatever. The drug and all the cell and tissue therapy that can be used. But you have all the products related to the recombinant technology, such as, for instance, nucleic acids, which are important for gene therapy, for instance, as I present you Tuesday. For the nucleic acid, also important for DNA vaccine or for oligonucleotides or plasmids that can be used for therapeutic purpose. And you have all the therapeutic proteins, monoclonal antibodies, and some of vaccine, recombinant or therapeutic vaccine, who are made of protein or part of proteins. So for general, so that's all the biological product. To highlight the problem related to proteins when we want to perform quality control, just remind you that protein are complex structure. You can see here a comparison between a MABS, which is 150 kilodaltons. His weight is 150 kilodaltons, compared to an aspirin, salicylic acid, acetylsalicylic acid. But I think aspirin everybody knows. It's 180 Dalton. So that's give you the challenge related to the complex structure of the protein when we want to perform quality controls. Okay, and to develop methods. And also why? So it's just the example I just highlight the maps, but there is many recombinant protein. But we can see here that the growing production of therapeutic MABs in the market. So that's why also, for this reason, it's mandatory to develop a rigorous quality controls methods dedicated to proteins and MABs. Okay. But that could be not. But there's not only MABs. It's one type of therapeutic proteins. So just remember also why biopharmaceuticals are complex, because they are manufactured. So as I said, as you know, of course, following biotechnological methods. And they have biological source, which involve living organisms as well as their component. And when for this biopharmaceutical, you have several. So, in fact, it's involved really complex media as well as complex biomolecules. So, for the following reason, first, They could be extracted from tissue, animals or human fluids. They can be issued from cell culture production. Prokaryote cell, if this is bacteria such as Escherichia coli, or eukaryote cells, this is mammalian cell lines such as Chinese ovarian hamster, as we have talked yesterday. They also can, it can involve transgenic animals or plants, as well as living organisms such as eggs, which is the case, for instance, for some vaccines. So you have many sources, and it increases the complexity of the problem. So I have already talked about that. So if you go deeper in the quality control issue, what is important, as I said, is the purity control has to be performed. For many reasons, for instance, for the recombinant protein, you can have in your media, once the protein are processed or produced, foreign proteins. You remember what I told you yesterday. When you produce protein, for instance, in CHO cells, CHOs, that will provide the desired proteins, for instance, monoclonal antibody or whatever. But you also have the protein of the host cell protein. So the protein produced by the cells themselves, which are not desired. So we have to, and even different processes are set up to be released, to remove them as much as possible. Sometimes some host cell proteins are still there. So we have to check whether they are completely removed from the media or not. You have also the related proteins, who can be also something detrimental if they are truncated or whether truncated or with small aggregates. We have also the DNA, the remnant DNA, the recombinant DNA, also viruses issued from, if we use a viral vector to produce them in the mammalian cell, or sometimes a contamination by another virus, and other. So that's why methods to ensure their identity has to be done, and most of them are the biological activity, the physicochemical property of the analysis, the dosage, the primary sequence of the protein, as well as the glycosylation analysis, and dosage, as I've presented already. So what kind of process-related impurities and contaminants can be found? As I just told you, cell host proteins. That can be also nucleic acid from the host cell genomes, or the vector, or that we'll call, and both of them, which is total DNA. That could be also issued provided by the cell culture, because we know that we use antibiotics mainly when we want to select the protein that has been properly transfected by the viral vector. You remember that in the plasmid, we add a gene encoding for a resistance to antibiotics. And then when the cells are growing, to be sure that they have been correctly inserted, they add antibiotics, and then it's help the researcher to select the good cell lines which are being correctly transfected. So you can have antibiotic who remain on the media, serum, bacteria, or whatever of the media. During the downstream process, you will have many steps: filtration, formulation, which can also lead to contamination by different agents. Even, for instance, contaminants which are due to, for instance, the filtration process. You filtrate through filters made of plastic. Maybe you can have some small component of plastic. which are extract. So it's extractable contaminants. It may have organic salt, solvents, also related to all the processes that are being employed to purify or to characterize, and ligands. So what kind of variant we can, for instance, when we talk about, if you come back to the proteins, which kind of variant we can find? Variant related to deamidation process. I will explain what is in deep later how the deamidation leads to the production of variant. Oxidation, which can also lead to the for instance, to a conformal alteration of the protein, as well as isomerization. So isomerization is more related to deamidation than, sorry. Alteration and post-translational modification, truncated form. This truncated form could be due to the chemical process, but also among the protein release, among the proteins composing the host cell protein, you have enzymes such as protease able to degrade the proteins, dimer aggregation, or uncomplete disulfide bridge. So here you have a small sentence extract from the ICH International Harmonization Guidelines. Who give some specification about test procedure and acceptance criteria for biotechnological and biological products. We know that why they say that, because they state that this statement exists because we know that, everybody knows that this production is a unique biosynthetic production process, and a drug substance could include multiple molecular entities or variants. For instance, you have some charge variant that can be produced after the upstream or downstream process on the drug products. That could be related to the deamidation, but that could be related to many other things. You also can have a variant in terms of glycosylation. Maybe you have more or less, 3, 4, 5 or sialic acid on the maps, for instance, and that will be responsible of charge variants. So some of them we have to accept that we have in the compounded drugs, for instance, in the infused bags for MABs, we have to accept that is not composed of all the molecules on this bag are not identical. There will be, one of them will be, BR3 sialic acid, the other one 4. Some of them we have glutamate and some of them not, we lose this glutamate, and etc. And it's difficult, we have to accept that. And also why we do that? Because once you produce your therapeutic MABs, when it became public and other company is allowed to produce their own MABs, like a generic for a normal drug, I don't know, for paracetamol, or for a lot of antibiotics or whatever, you have now, they are in the public domain, we say in France, I don't know how we say that in Spanish, and other companies are allowed to produce generic, which is the same identical compounds, but it's not the principle. But for the proteins, we don't call that generic, because we know that it's not possible to produce a generic, it's called biosimilar. So that means it's a molecule who exhibit similar biological effect. Okay. So also, when these molecular entities are derived from anticipated post-translational modification, they are part of the desired products. That means exactly what I mean. We accept that there is a variant, acidic variant, sometimes hydrophobic variant, and so on. And when variant of the desired product are formed during the manufacturing process and storage, and have property comparable to the desired product, they are also considered as product-release substance and not impurities. So that means we accept, in general, we accept that all the molecules are not identical in the same formulate in the same drug product. So I have to, so just for, as it's highlighted here, sorry, sometimes when the drug, you have when you produce the recombinant protein, you have the upstream process, the downstream process, I will explain it. After the first part of the downstream process, so harvesting, purification, polishing, we obtain the drug substance, and after its formulation, it's called the drug product, okay? So it's different, and the drug products, I said, what all the molecules even are not identical in the drug product, as well as the drug substance, in fact. But so here, in this schematic representation, you can find all the sources of chemical and physical stress that arise from the drug product manufacturing process. Okay. So we can see that light source is they are submitted to many stress, so light source, which can induce oxidation, and when a protein is oxidated, that could lead to a modification of its conformation, and then the biological activity will be different. There can also be, sometimes when they can be submitted to also variation of temperature, which can have an impact. Most of the time the temperature is 37 degrees, but sometimes they have to modify the temperature during the process, and that will be a source of stress for the protein. We know that when they are submitted to higher temperature, this could lead to a modification of the conformation. Also at the clinic stages, when they are reconstituted in perfusion bags, the perfusion bags, they could be traveled inside the hospital and submitted to light stress. They also, it has been demonstrated that when some therapeutic protein, when they are administered to the patient, they use a syringe that give a pressure during the administration, which could also lead to a modification of their conformation, and also and they show that for therapeutic mAb, that could induce, in the fragile ones, all of them are not fragile like that, that could induce formation after that, formation of small aggregates. So that could be something. During the development, at different stages of the development, sometimes they perform some experiments with, they can perform, some steps require cryoconcentration, so freeze-thaw, and that's not good for the protein. So they have to check what happened. Also a lot of mixing, agitation, or shear forces, which can also be detrimental for the proteins. And the last thing is even at the hospital, sometimes when you have they collect the final products and they reconstitute at the hospital, and it's sometimes difficult to establish a proper quality control at the hospital. So in a lot of hospitals, not of them, the control is made, is just one person who's preparing the drug products, formulate the drug produced in situ, and the other one is it's a visual inspection, but there is not no rapid method to perform a quality control at the hospital stage easily with the proteins. So here is all the summary of the and they have to take into account, I didn't the different steps here, which could be it's important with the time at room light, because of the light exposure, the time out of refrigeration, sorry, because of the modification of the temperature, the freezing, and also what I said, the dilution at the clinic. So just in this table, it's a summary of just what I presented here. All the different stages where all the source of stress during the drug product manufacture, but I already said during the process, during the formulation, during filtration, filling because of the pumping and the shear stress and the interfacial stress. We know that when the protein are in the infusion bags, at the interface between the liquid phase and the air, protein just at the surface can undergo modifical conformation. It's just, I will develop anyway later, but what all the different process. So here I develop a little bit all the factors who can leading to the degradation or inactivation of the proteins. So the chemical factor that could have an impact on the stabilizing forces of the native structure, such as hydrogen bonds, hydrophobic interaction, electrostatic interaction. So that's the case with the detergent, organic solvent, and chlorotrops. Some of them establish complex with amino acid or catalyze reactions, such as metal ions. You have biological factor. Sometimes protein, co-purified proteolytic enzyme, as I said, which who are produced by the host cells, unfortunately. Carbohydrates, phosphatase, sometimes deglycylation, which reduce the resistance to the protease. So, also factor leading to the degradation or the inactivation of the protein, such as all the physical factors, so the mechanical and agitation stress. So the air bubble produced, as we know that at the interface of air and liquid, the protein are fragile. Gas-liquid interface, also denaturation of the protein. All the temperature freeze drying, which affect the stabilizing force of the protein, and so have an impact on their folding. The pH, extreme pH, they don't like that, which affect the ionization. Oh, you are still, sorry, still word in French, not sorry, but affect the stabilization of the amino acids. We know that asparagine or glutamine can be deamidated, and we have also the new aminic acid for the glycoprotein, who can be also affected by the pH variation. So for the deamination, we know that especially asparagine is prone to deamidate, mainly at high pH, and aspartate could also isomerize at low pH at that time. And we have also a problem of oxidation that could occur under light, under peroxide, atmospheric oxygen, or redox sensitivity. That could be related to the metal catalyzed oxidation, to the photosensitivity. You can have oxidation, mainly it's methionine and tryptophan who are oxidized, or also due to the peroxide. Okay. So deamidation occur at different levels. That could occur during the cell culture, so during the upstream process. It could also occur during the purification process, so downstream processing. It also could occur during formulation, so and during the storage. So at many stages it could occur, and when the deamidation induce a loss of amino group that could lead to the formation of charge variant. Also here the isomerization of the aspartate from aspartate to isoaspartate which can also, and both of them could impact and also isomerization could occur during fabrication, development, formulation, and storage, and also could impact the protein conformation and stability. Here is all how the different process leading to the oxidation of the protein through two amino acids, so methionine and tryptophan. And why there is a structural damage? Because oxidation alters the protein tertiary structures, which lead to conformational changes. They could, and thereby unfolding, and at the last stage, aggregation. Oxidized protein often show a decrease of the biological activity or potency. And as I said, it's increased instability by accelerating degradation and aggregation, and increasing thereby immunogenicity, which lead to, as I said yesterday, I think, the production of drug antibody directed against the therapeutic antibody or the therapeutic protein, and so decrease his therapeutical efficacy, and also generate unwanted immune responses. So what is the cause and what is the risk? So to summarize what I said, all the cause and risk factors for protein production could lead to the intrinsic property of the protein when they are degraded, when they are degraded. So degraded, that means truncated or aggregate. Also, during the time and temperature exposure, and we know that proteins are considered as sort of polymer of amino acids, and they are not. They are weakly. Some of them exhibit a weakly stable thermodynamically, which run them prone to degrade. And also we know that they are sensitive to the exposure to interface air and water, mechanical stress, such as shear stress, for instance, during the injection to the administration to the patient, and ambient light. So that's why we know that all the proteins such as monoclonal antibodies are for all the drug anyway. Critical quality attributes must be defined, and which is really important. And what is a critical quality attribute is defined as the physical, chemical, or biological or microbiological properties that should be within an appropriate limit, range, or distribution to ensure the desired quality. For instance, if we know that for MABs we have dimers, a critical quality attribute will say no more, the dimer should not exceed, I don't know, 0.01% of the drug products. Okay, that's a critical quality attribute. Truncated form should not exceed 2% of the drug products. That's defined range. Anyway, they were presented here anyway. And here is and the list of potential quality control attributes comprise product variants and process-related impurity, which may potentially impact molecule mechanism of action or mechanism of toxicity. We know that when we have aggregated form, it's decreasing their biological activity and generate toxicity, and which lead to safety issues. So here, for instance, you have that is the list. We don't see it. I try to a list here that's for MABs, monoclonal antibody, a list of variant critical quality attributes. So, for instance, the size variance, that means the high molecular weight species, that is related to the small aggregates. So they have to be, they do not have to exceed designed range, okay, not more than 1% or whatever. It's the same for low molecular weight species, that means truncated. They must not exceed a normal range. It's true for as well for charge variant, acidic variants. Deamidation in the region involved in the biological recognition of the target. It's also in this region, but it's also true for in the other region involved in the what we say, for instance, of the SE region involved in the effective activity of the MABs. We have also charged a basic related variant who lose one aspartic acid or isomerization of aspartic acid, also a loss of lysine in the C-terminal lysine, which lead to a variant. You have also oxidized variant, glycosylation. We know that there is an issue for fucosylation, galactization, imano-sialization. And for structural variant, for cystinic form or sequence variant, or for the protein structure as well. So there is all the list of that have to be the manufacturer have to stick to this quality control attribute. I think I can skip that because you know that all the protein transformation. So why is that? So now you have understood. I think that especially when you talk about therapeutic protein, they are generally they differ by they are complex by their size, their high structural complexity, so you know now, and also their heterogeneity. When I talk about variants, for instance, you have one glycoprotein, but is related to a mixture of glycoform, which could generate different molecules in the same drug product. So the protein, the protein are submitted to many post-translational modifications, such as glycosylation, disulfide bridge, lipid onshore, acetylation, and so on. But it's not phosphorylation. The glycosylation is something important to consider as a major post-translational modification, which has a big impact for biopharmaceutical. So we have two types of glycosylation. N and O glycosylation. And why they are so important for the... So that's the different type of glycosylation that you can find on the therapeutic proteins. And why they are important for biopharmaceutical? Because they compare to the drug products solubility and stability. They are also important in their folding, also in the process involving cell recognition and especially binding on receptors. Their bioactivity, their pharmacokinetics, because we know that when they are less glycosylated, the time, the in vivo time is reduced as they are captured by the liver cells. Their elimination as well as their antigenicity. So here, well, I think we have done said that. I think now you have understood all the different steps, which is very important. Everything has to be considered for monoclonal antibody in terms of development, upstream process, downstream process, bioanalyzed for the quality control. and which is summarized here in this figure. But I think here, for the, I come back to the therapeutic monoclonal antibody, and here is all the modification that a monoclonal antibody can exhibit. He can his oxidation could be modified. It could be isomerized. The modification of his glycans, modification of his lysine, deamidation, fragmentation, aggregation, and also form particles. And here, for instance, you have the impact of this variation on his on the MAB's physicochemical properties. So here you see, for instance, when by deamidation, modification of the deamidation, the removing of C-terminal lysine or glycation, or addition of N-terminal pyroglutamic acid, that leads to the generation of charge variants. By oxidation and disulfide bond scrambling, that lead to the generation of hydrophobic variants, and glycosylation could lead to the generation of hydrophilic variant as well as charge variant, as I said before. So when we want to characterize a MABs, we can do that. So here we have, so for the quality control attributes, as I told you, we cannot have exceed a certain range for charge variant, small aggregates, and so on. So to check all this feature, we have to characterize the MABs once he's produced. So we have different way to analyze globally a MABs at different level. We can analyze the entire MABs. MABs could be partially digested, with its middle down by analyzing its middle down fragments, for instance. So, for instance, after IDES, for instance, that's enzyme who cut the MABs here, and we have in one. So he's divided in two. He's cut in two with a Fab in one side and the FC2 in the other side. You have also the papain who can separate FAB and FC, but not one. Or DTT who cut in two, but with the two light chain and the two heavy chain. And at the end, we also with enzymes such as trypsin or lysine, it's the bottom up with by generating a lot of peptide to analyze them by chromatographic methods such as liquid chromatography coupled to mass spectrometry. So to analyze the MABs, you have different methods that could be done to check, and all these methods will allow to check the purity and the identity of the MABs. Okay. If we come back to the quality control, you have chromatographic methods, electrophoretic method, mass spectrometry, and spectroscopy method. I will not go not detail those one, but the most simple one. So for the chromatographic method, ion exchange chromatography, for instance. So here is summarized all the charge variant that MABs could produce. So you have different acidic variants. So when the normal average amount of variant in MAB will be acidic variant between 5 and 30%. The main peak is between 25 and 90%, and basic variance 3-4%. And so when the first method is the ion exchange chromatography, you know this? You know chromatographic method or not at all? Or do I need to develop? Do I need to explain or no? Yes or no? No, they know. No, it's for the student. And, no, do I need to explain or not the general principle or not? It's just a question. You can say yes. It's okay for me as well. No problem. Just to know. Yes? You want? Okay. Ok, just a drink et je vais. So just before explaining, presenting the ion exchange chromatography, just remind that what is isoelectric point, but maybe you know that. I don't need to explain that. Ouais, c'est bon ça, ok. I will. So just the general principle of ion exchange separation. So this type of retention is governed by electrostatic interaction between the opposite charge on the stationary phase and the protein. And the selectivity is dictated by the difference in protein charge. And elution is allowed mainly by running a salt gradient, but it could be also a pH gradient, depending on the methods. And so just to show here the principle of the method here, then the protein are the analyte to be separated are here, and then they are the elution with the buffer. The buffer is is percolated through the colon and will elute the buffer will elute the analyte regarding their their global charge. So just So here for the cation exchange, the mobile phase, the stationary phase is charged negatively. The analyte is the analyte to be the protein to be eluted will be globally positively charged, and the buffer here the buffer will elute the protein by the buffer positively charged. The analyte composing the buffer positively charged will elute the protein, and the protein will be analyzed and by the stationary phase. It's the other way from the anionic exchange, where the stationary phase is positive and the analyte, the protein to be analyzed is globally negatively charged. And you have several types of stationary phase, what we call a strong cationic exchanger and a weak cationic exchanger, depending on what the analyze perform. We skip, so I think it's okay. We're not saying. So just to remind, when you change the mobile phase pH or when you go to the salt, when the salt concentration increase, that allow an adjustment of the compound retention, and increase of the salt concentration provide a reduction of the retention, and the salt gradient is mainly preferred in most of the time. So just here, what the type of profile obtained when mass is analyzed by ion exchange, you can see here. So you will obtain. So here is the profile. You have the salt gradient. You have for the protein you have the main peaks. On the right the basic variance, and on the left the acidic variance. And that this profile allow to establish a sort of identity for the proteins. Okay. And that in this case a strong cation exchange column has been done. And we can see here. They compare the profile obtained for MABs when they are normal MABs and MABs under oxidation, under forced oxidation. So they perform a forced oxidation by submitting the MAB by adding on the MAB preparation a solution of hydrogen peroxide will generate oxidation of the methionine and the tryptophan, methionine mainly in this case. And as you can see, for instance, for the monoclonal antibody A, you will see the modification of its profile. Here, you can see that the profile is modified. For the MABs B here, so you have the analysis of the main peak. the basic variant, the acidic variant, and you can check that by be submitted to hydrogen peroxide, you can observe a modification of his profile. So you can see by using a chromatographic experiment that the profile is modified. So that means that oxidation, the maps have been submitted to oxidation, and that could be detected by using by his analytical, his chromatographic, by his chromatogram. Okay. Is it okay for everybody? Oui, oui, toi je pense, je sais. Toi, c'est normal. Toi c'est normal, but c'est okay, okay. Okay. So here I skip this one. So then the other one is what we size exclusion chromatography. So that's allowed the analysis of protein size variant. I assume that is well known for most of the students. The principle, I think, is known for everybody knows the principle of size variance, I assume. But you can say no if you don't know. Do you know the principle of size exclusion chromatography? Oui. Yes. So here I can skip. So that, for instance, that could help to identify. First, it could be done to identify the protein. You can see here in column A the analysis of a mixture of different proteins, and there will be the retention time varied according to their molecular weight or to their size. And if you analyze MABs, normal MABs, you will MABs one you have the control just here in this is a I'll say I come back. Here this method has been applied to MABs, and they compare control MABs and MABs submitted to stress. And you can see here MABs have been maintained 3 months at the same temperature. Okay, so there is a control MAB here in this chromatogram in black, and then so here is the main peak, and okay, it's the main IgG, and it's normal, and everybody's okay. Then they took the same MAB, and they maintained the MAB. They conserved the MAB during 3 months at room temperature. Okay. And they see what happened to the MABs and to its stability. And you can see that's the chromatogram in red, that on these MABs you find 2 fragments. You still have a small aggregates, and you have 2 fragments. So that means here, because the first MAB was a control one, but you see here in this first peak, you see small aggregates, okay? But in this case, the quality control attributes say, okay, this is less than the 0.1% of the MABs who is aggregate. It's okay. The product is still a drug product that could be administered to the patient. When they submitted this MAB, they keep this MAB 3 months at room temperature, we can see that fragments, you can observe those fragments. That means the MABs is truncated and degraded. So here, okay, we can see that by this, in this condition, we cannot keep the MAB 3 months at room temperature. That's not good. I can't keep the MABs and administrate to the patient 3 months later. It's not possible. Okay, so that could be. It's an important issue for the hospital. You know that MABs have to be prepared immediately and administered immediately. You can't keep it at room temperature during 3 months. In this case, it's they control the MAB during the process of production. So the first MAB was a MAB control, properly processed, but you can see anyway in MAB 2 that you still have, you observe here the small aggregate, but the level is important. I'm not sure here that it will be okay for the quality control test, okay, issue. And then when they keep it, they change the process and they control the MABs after this new process, and you can observe that the peak corresponding to the aggregate is increased. So that means that here in this case, the process to produce the maps was not good, okay? You can have here control, we show, okay, but that's, here we have a degradation. So in terms of purity, it's not good. In terms of identity, we know that this map, the retention time is, I don't know, he's here, 2:15, I would say, 2 minutes 30. So that could be considered as an identity sometimes. And the profile is okay, it's him. But what we know that even if the identity is okay, it's not good for the purity as the level of aggregate increase, okay? And so here is, they performed the same experiment, but just here they changed the column, okay? The column used to analyze the maps is different. And what you can see, is it I can't say. The profile is not exactly the same, but anyway, the result here, the result is nearly the same. That means that with both columns, the interpretation is similar. That means that the aggregation proportion of the maps increase. Okay. So here is but that's why that show you why here what is important. What is interesting? It show as well. You can try different things. By using the column, even if the results are the same, the sensitivity is not exactly the same. So they have to. It's important to also perform development to ensure that the analytical method obtained for the maps analysis is accurate and will lead to the good interpretation or the most accurate interpretation. We know that for size exclusion chromatography, that sometimes we could for size exclusion as well as for size exclusion chromatography, we know that there are maps could also interact with the stationary phase during the analytical process. So that's why they have to develop and to develop another it's at the end of the first hour. I start to be a little bit tired. No, just to say that we have to be sure that the mobile phase is perfectly accurate to avoid misinterpretation. So that's why they have to be the mobile phase has to be investigated as well in research, in terms of research, to check whether or not the interpretation could be modified. And you can see here, in the first case, the mobile phase was similar, but the stationary phase was changed. Yes. Here, the column remained the same, but the mobile phase has changes. Just to check something. No. Okay. So here they compare, in fact, they analyze the maps. So you find again the monomers, the small aggregates here, and the truncated form, and they modified the concentration. And what is important is to check, because what they want to be sure that they did not avoid anything. For instance, if the separation between this fragment and the monomeric peak is not sufficient, sometimes this one could be hidden. Okay, and if it's hide, that the interpretation could be wrong because you said, okay, but there is no truncated form of the maps. So it's important to be sure that we can identify all the species present in the drug products that analyzed here. Let's just open it. And so I will not describe in detail the chromatogram here, but what is important to look at is here the different the proportion of the proportion of small aggregate on the species analyzed here. And we can see that depending of the mobile phase, we observe variation in terms of measurement of small aggregate, because here, for instance, when it's a mobile phase with sodium phosphate or mobile phase with ammonium acetate, you can see here, for instance, for this map, that the proportion of small aggregate is not the same. Okay. Here is so that could lead to that could be important in terms of quality control attribute, that the good proportion of map could be has been determined. Otherwise, you can that could be that could lead to a wrong interpretation. We can see that here as well with ammonium acetate 20 millimolar. You can see that the proportion of the percentage of small aggregate is really different compared to the 3 other mobile phase for the same column. Okay, and it's the same each time. That's why the and on the other way around, when they look at the peak tailing, so that means the let's say the width of the main peak. Okay, it's okay. Attends, ouais, non, mais c'est plus facile à expliquer comme ça, je pense. Oui. For the peak tailing, so that means the width of the main peak, you can see that it's higher most of the time for this mobile phase made of ammonium acetate at 20 millimolar. That means that when the peak tailing is important, that means that you can miss a truncated form or a small aggregate because of this this large peak who hide species that are here, but you cannot see them. So that's important, and it's really important. Maybe so. I don't know if you like. I'm not sure you are fan with analytical chemistry, but it's really important anyway, because it's really important to determine precisely the percentage of small aggregate or truncated form, as they are really important in terms of quality control attribute. That means if you don't respect, as I said, you have to stick to the control quality attribute, because it's important in terms of patient, and then with this, you need to know exactly if the drug product that have to be administered to the patient is good in terms of quality control. If it's not good, you misinterpret and you administer to a patient a drug product containing a high level of small aggregate, and that could be detrimental for his health, because it generates unwanted immune response, and it's generated antibody directed against the drug, and so it decreases the therapeutic efficiency of the maps. So that's why it has to be precisely controlled. So so to conclude with the size exclusion chromatography, there is several advantages, because it's a widely accepted technique by the regulatory agency, and also it's a quantitative set method who can validate the precise percentage of unwanted species, which is important for the quality control attribute. Disadvantage, as you have seen here, sometimes the resolution is limited, especially for certain proteins, and sometimes it's difficult to discriminate between close protein close interval of molecular weight. And also the colony is expensive and have a short lifetime. I think also some quality control can be performed with RPHPLC. I can skip maybe, because you can see here the different by RPH principle. I think you know as well this method. Ils connaissent? Pas besoin de. Ok.
Tu peux faire un test pour eux.
Ah, c'est demain. C'est pour demain. Tomorrow. I haven't. No question today. Sorry. But here you can see. You can compare 3 chromatograms of MABs, normal MABs, MABs submitted to acidic stress or basic stress, or MABs submitted to oxidative stress. And you can compare the profile obtained. So in A is the quality control, in B is the acidic stress, and MABs submitted to basic stress. And you can see here that the modification of the profile, especially in the main peak. Okay, so that's so then, of course, here in this case it's normal because we have submitted MAB to stress. So of course we are going to obtain the chromatography. The chromatogram will be different between the normal MABs and the MAB stressed. But what is important is to keep in mind that in the pharmacy at the hospital, before the MAB administration, they have to perform a control to check if that the MAB administered to the patient is in a good state. Okay, so then that could be something to do, to analyze quickly the MABs before the administration of patient, and they can see that okay, that's the normal chromatogram. If the MAB that the reconstituted MAB that has to be administered to the patient start to look like that, they say, but okay, this MAB has a problem, so maybe we are not going to administer this MAB to the patient. So it's really important in terms of quality control, anyway, to check if there is impurities, but also in terms of safety before administration to patient. So here in this case, it's less a quality control in the in-process, okay? It's more a quality control at the hospital before the administration to patient. But it's a quality control anyway.
Can I ask a question?
Oui.
You know, so now there are forms of antibodies that are administered by subcutaneous injection, and patient, I'm not sure if the patient are administered at the hospital or at home.
At home.
Do you think, because if it's administered at home, how is it possible to make a quality control before you?
No, you can't. Anyway, for me, this method is not suitable anywhere at the hospital because they need. It's a good way until the control, but it's too long for me. The only thing I can see for the patient at home is difficult. Anyway, the thing that they can do is to, if a proper method is settled at the hospital, because the delivery is by the hospital, right? If I remember this kind of way.
Sure.
I think so. But anyway, they have to analyze the MABs, the same one in the same condition, and you analyze and you to check the profile. And you say, okay, in this batch, probably it's okay. We can give it to the patient, and he can self-administrate at home. I think it's the only way for me. But it's true. It's a good question. But usually, and also for the MABs administered subcutaneously, the concentration is very high, which is also can be a problem. And also a lot of constraint. But anyway, they know that, and also it depends on the MABs, because some MABs, such as infliximab here, are really prone to aggregate. But he has a sort of cousin which is called adalimumab, who has the same mechanism of action because it's directed against TNF-alpha, and he's administered subcutaneously as far as I know. And but he's more. But all the tests that we have performed with we compare infliximab and adalimumab is less prone to aggregate compared to infliximab. So you have many factors to take into account. I agree.
Ça s'arrête là.
Oui, ok.
Ça s'arrête là. Je veux dire, si pendant l'analyse il trouve des produits de dégradation.
Oui.
Ça...
Ah bah oui, non, il ne l'administre pas aux patients. Mais,
oui, d'accord.
Oui.
Mais après, l'échantillon.
Oui.
Et ça revient au laboratoire pour identifier les produits de dégradation.
Alors,
Il y a
je ne sais
la
pas, ça dépend.
base derrière,
Parce
après.
qu'en fait, à l'hôpital, switch in English, non ouais, ok, vas-y. Ouais.
Pregunto si eso, el análisis se detiene ahí, ¿ya? Si es que después de encontrar, por ejemplo, en una muestra productos de degradación, se detiene ahí o vuelve la muestra, por ejemplo, al laboratorio, no en el hospital, para analizar qué es lo que es ese producto de degradación. Eso. Así.
Okay. So, but at the hospital, not all the hospital has this type of analyze, of control, because of the MAPS complexity. Usually, when you visit, we visit the hospital, the pharmacy, what you call the pharmacy center unit at the hospital, the place where all the drugs are prepared before sending to the patient and administered to the patient. When they reconstitute the drug, they do it in the pharmacy central. Within the small molecule, they are really rapid control. In 1 minute, they know if the MABs is okay, or the product is okay, not the MAB. But for the MABs, as it's more complex, they have less method. They have some method, but usually, if it's not, they have some method, but it's not very accurate one. So that's the problem. And anyway, when you prepare a MABs, and if you still have MABs remaining, the patient did not use everything, you never keep it. You just keep it, but you will never reuse it. They don't reuse most of the time the MABs. That's why also they are doing a lot. It's difficult now to reuse MABs because they don't know what is exactly their property. And I'm not sure that, because also you have a lot of, when it's reconstituted, and if they find something, then maybe they will, what they will do, probably, if I remember what they said, if they identify something, you keep it and you don't give it to the patient, of course, but you take another batch and you do it again, and to just check it's not a batch effect. And usually, maybe that could be done. But it's really controlled precisely. When you reconstitute a bag, it's really a visual control, and the person is filmed, and you can. But okay. So here we can see for the. So as I said, that's the first step toward the control before the patient. But here, in terms of, especially when for the big hospital. Even a control for a chromatographic method of MABs, for me to obtain a peak in 8 minutes is rapid, considering the techniques, the analytical techniques. But for the hospital, it's still too low, probably. But but we can see by this method what is interesting is that they can they can compare the profile, and check by controlling the profile what happened if the MAB have submitted to stress or not. And for instance, in this case, you have a stressed MABs, normal MABs, the MAB submitted to thermal stress and UV stress, and you don't see any difference. So that means, okay, these MABs anyway, probably that could be okay. So, because they know which MABs are. But anyway, it's difficult because even you analyze the MABs before the administration of patient, the modification of the conformation could happen during the administration of in the perfusion, in the syringe. And then what can you do? It's difficult because you know that you're going to administer MABs degraded, and we know that there is problem because we have, after MABs administration, they observe immunogenicity. So it's a problem anyway. Up. So for HPLC, so sometimes HPLC give you an indication, but anyway, as the conditions employed are harsh, the mobile files with acidic additive, with organic modifier, and sometimes, but not all the time, at elevated temperature, they are not completely sure that the MABs, normal MABs, is not himself degraded. So the next method is hydrophobic interaction chromatography. Oh, here, so the different, so it's also, we can use MABs, but here the chromatography is performed with a high level of organic solvent modifier, and there the elution will be, so the less will be performed in this order, the less hydrophobic molecule will be eluted first, and the most hydrophobic molecule will be eluted later. So and in fact, just you know the principle. I don't need to explain the principle of hydrophobic interaction chromatography. Is okay for you? Okay. Yep. So here, for instance, you can see a difference of profile, MABS profile in terms of when they are a normal one and the antibody incubated at 40 degrees, and you can see here differences. Difference of the profile obtained by hydrophobic interaction, and you see here two other hydrophobic species, less hydrophobic than the main peak, but hydrophobic variation. This technique is able to also separate MABS aggregates. So you have here the MAB variant. So that variant is hydrophobic variant here, and here it's here you can see aggregate. So a different is also able to separate different variant, but hydrophobic one. But you can see here that the MABs have been oxidized. And the difference with the different profile with the oxidized MAB. So here is the normal MABs profiles, and here when it is submitted to oxidation by hydrogen peroxide, you can see a modification of the profile. So that shows aussi that the highlighting the modification of the degradation of the MABs. And again, so the comparison, the evolution of the profile when MABs are being submitted to increase of the degradation, and we can see. So here it was a MAB similar to biosimilar, and we know that the percentage the oxidation is also a critical quality attribute, as I show you at the beginning of the presentation, and. You can see here the modification of the. You have here is the control MABs, and by increasing the time submission to oxidation, the time exposure to oxidation, you can observe the modification of the profile. So that's the profile, and you can see that here there is also an impact of the MABs. So maybe I skip. I think you have understood now that the degradation modified the profile of the interesting part. The interesting part of the chromatography is the fact that by submitting the MABs to different stress, and usually they use oxidation stress or light stress or whatever to reproduce as much as possible the condition, the upstream or the downstream process condition, and also the condition, the condition where the MABs is submitted, where the MAB, the condition for MABs after his reconstitution. So, what is the main stress? For MABs, sometimes acidic condition, light exposure, oxidation, and temperature. And what we saw here is that by analyzing maps by chromatography, you obtain profile, and this profile is modified, as you can see, when the map is submitted to stress. So here you have an example with oxidation, and a lot in the publication we'll find a lot of work with oxidase map, as one of the most stressed, is the most easier stress to mimic. Okay.
Can I ask a question?
Ouais.
You showed before that size-exclusion chromatography can be used to characterize the variation on maps structure. Can it be used also for purification?
It is used for purification, but at larger scale. Yes, it's used as well.
Because you can think that if you check quality control, you see there is a problem, so you can select the one that is correct.
But size-exclusion chromatography will allow to collect the main peak, but you don't know. You see a profile, you see the main peak, but you don't know what is the state of the species inside the main peak. That's why sometimes they like, when you want to go deeper in the analysis of maps, you add a second dimension. So you can do a first dimension with SEC and the second dimension with IEX or whatever to see the impact of the profile, because it happens also that you can have modification, conformational modification, but you don't see it in this. It's all the time an orthogonal situation. We need an orthogonal methods to carefully characterize a maps or a protein or whatever. You need several methods. So I can skip. I think you have you understand now that the interest of the analytical sciences, and that's allow me to, if you want to go deeper in the characterization, because as we just said, thank you for your question, Nicolas. That gives me the perfect opportunity to switch to shift to the other part of the presentation. That, for instance, in the monomeric peak on SEC, for instance, I see a monomeric peak, so I can say, okay, we have one peak, no truncation, no aggregation, but I don't know if under my monomeric peak there is no conformational modification or no modification that is not highlighted by this method, by SEC. That could happen. Also, sometimes the truncation is small, you don't see it, but still it's important. So that's why now the 2D LC approaches have been proposed. Just to illustrate it here, the interest of the second dimension. Ça me donne soif de boire comme ça. Sorry, just. You can see that you can do what we call multi-hurt cutting. That means, for instance, you have the first peak here, and you can, in the first dimension, but you can then analyze it in the second dimension, for instance. So it's maybe better illustrated here. You have the first dimension, you take this peak, and you can see that you can find other peak here, as well here. So you can and you can combine different possibilities. So you can do this here. You have this first group, which is not well separated. You go to the second dimension, and you obtain a better resolution between the third peak. That could be the first purpose for 2D separation. But you can do also, you can combine two different type of methods. As here you have the first the group of three peaks, and you improve the separation. But you can do also a thing that you can analyze just one of the peak, and you can check to see inside this peak by another method, an orthogonal mechanism, another peak. Maybe I'm going to skip this one because it's a little bit complicated. So that's the model, but I think I'm not going to go deeper in this example because it's very complicated. And so just here to say that you have orthogonal methods that can be combined to improve the separation and the analysis of one peak of interest, for instance. And then I can switch to the electrophoretic methods that could be used as well. So here is a summary of the all electric method that can be used to analyze the identity and the impurity as well as the degraded form of the proteins. So here most of the capillary electrophoresis, all of the electrophoretic method rely mainly on capillary. And why this method raises a lot of interest is that you can analyze them. You can use very small volume of samples. For chromatographic method, you can analyze samples around 10 microliter, which is small, but really, for instance, in a capillary electrophoresis zone, the volume of the sample analyzed is 14 nanoliter. So it's really a downscaling, an important downscaling. And you have as well, you can use capillary gel electrophoresis. We allow the separation of the protein regarding their size. Capillary isoelectric focusing analyze the charge variant of the protein. So it's related to the difference of related to the pi of the different charge variant, because we know that each charge variant exhibits a different pi, isoelectric point, and capillary zone electrophoresis, who can allow the analysis of different of heterogeneous of different molecules exhibiting charge heterogeneity. So I think capillary gel electrophoresis, you know the principle. I think you don't. No? Yes? No? Oui, connaissent? Yes. Plus ou moins? Okay. Well, so capillary gel electrophoresis, so it's a sort of, as I said, a modern version of the SDS page. It's the same. So you fill the gel with a gel polymer matrix. You add exactly in the same way SDS as the page. The SDS will interact with the protein. So you remember the SDS, you have a hydrophobic part and a charge, okay? And the hydrophobic part of the SDS will interact with the hydrophobic part of the proteins. And so the protein will be globally negatively charged. So we suppose that when we applied an electric field, they will migrate with the same electrophoretic mobility, because we consider that the ratio charge mass is similar for all the protein. So they will be separated regarding their size. Okay. And so that's, well, I skipped this part, but it's really exactly the same. It's a gel, and with SDS, and we apply the electric field, and there will be a sieving of the protein through the polymer networks, and they will be separated in the increasing size order. So it's as usual. So I can skip here, and you can see here an example with therapeutic monoclonal antibodies. I hope you will not be fed up at the end of the lecture with the therapeutic monoclonal antibodies. So here we can see that the map has been reduced with 2-mercaptoethanol. So they can be separate. The heavy chain of the map will be separate from the light chain. So you obtain 2 peaks. And here they compare the profile, the electrophoretic profile of the electropherogram, we said. As we said, chromatogram for chromatography, we said electropherogram for electrophoresis. And here is the profile, the electrophoretic profile or electropherogram obtained for MAB initial sample. And the MABs has been also submitted to degradation, so it was here, it was kept during 6 months at 25 degrees, and you can see small degradation here. So we see that the MABs is able to highlight degradation. So that's when the MABs was reduced to separate light chain from heavy chain. Here is the non-reduced MABs, and you can see that here is the initial MABs and here is the degraded sample, but without the reduction step, we can see that the result is not ideal. As for instance, probably we have something here, an increased peak, but really you have to put really your focus a great deal of attention to notice it. So, so that's highlight the fact that you have to carefully study your protocol, and here you can see that by reducing the MABs, you can highlight more easily the degraded part of the MABs. But we have also, it's also all the time a compromise, because we know that the MABs treatment, I mean the reduction of the MAB could also induce a degradation. So it's not, the method is not probably completely ideal. That's another development with MABs. So they compare MABs at the different stages. So they are all being reduced to separate the light chain from the heavy chain. And they compare the MABs. So that's the MABs who was frozen and defrost. And here the MABs after storage at 25 degrees during 104 weeks, I don't know why. So we can see here an additional, an extra peak. And here is MABs after storage at 25 degrees. So it's the same MABs, but submitted to a treatment with PNGASF. You know what is this enzyme? It's a MAB who allow who deglycosylated the MABs. Okay, so it suppressed all, because you know that the MABs are glycosylated, so then you suppress the glycosylate, the glycides, and then the carbohydrate. So and so you can see a shift of the peak when the MAB is deglycosylated. So what is interesting here is they show that so you can identify a degraded MAB, but you can also identify. You can also evaluate the level of glycosylation, and therefore a level of post-translational modification of the proteins. Okay, so there is two things here: the degradation and the glycosylation. So here they try to. As well, it's all the time really important to evaluate in deep the condition to be sure that to avoid any misinterpretation, as I said, as it's related to quality control. So here they use a different type of gel to evaluate the impact of the gel by mixing on different proportion of monomers and crosslinker, and you can see here. So here is the MABs are reduced, and you can see 4 peaks: one peak for the protein, for the light chain, for the non-glycosylated heavy chain, and the glycosylated heavy chain. So you can see several peaks here. And you see that by modifying the gel composition, what is important is to see that the resolution between the peaks are different. And here what is interesting is that they have been able to, for the same chain, to separate properly the non-glycosylated heavy chain and the glycosylated heavy chain. So that was really interesting as well. So here you can see two things. Here they are also able to, by this using this method, they are also able to differentiate. So the MABs is not reduced, has not been reduced here, and you can see that they can separate non-glycosylated from intact MAB, and they also compare the different proportion of monomer versus crosslinkers. The resolution is not so different, but what is important here to see is that the migration time is not the same. So there is an impact of an important impact on the migration time. And here, and so here they try another type of gel to see it, to see the influence of the separation, the MAB separation, and you can see that they can. So really, they can, all the species analyzed are here, and you can see that they have been able to analyze all the species on the mixture. And what is important to see is to observe is that again the proportion of the gel is important. They have varied here the percentage of dextran versus borat, and so we can see here the impact of the gel composition on the separation of the different species. So here the advantage here is that they were able to set up something very easily with a very small small volume of sample. They can increase the resolution. They can play with the different proportions depending on what they want. If they want to go fast, if they want to separate better different type of species. So it was really an interesting, something really interesting. It's also important to they also insist on the fact that this method is highly automated. So you are just to add your sample. It's not to press a button. It's not exactly that, but it's quite really easy to use it. And the limitation is the low sensitivity. And sometimes the binding of the SDS is not completely the same for all the molecules. It's true, but that's the limitation. But anyway, it's important to notice that for the MABs, the concentration of the MABs, the therapeutic MAB is quite high usually. So it's the low concentration is maybe not a problem here. Pourquoi j'ai ça? Sorry. Here. So the next, je sais pas ce que j'ai fait. I mixed it. The next type of. Après, je crois que j'aurais fini. So the next example I want to show you is ICEF. So it's exactly the same. You have here the gel SDS was the CE SDS was similar to the SEC mainly, more or less. Here the ICEF is something quite similar in terms of principle to IEX, ion exchange chromatography. That you obtain a profile here. You can see so here how it's work. So the capillary is filled of ampholyte, and when the electric field is applied, a pH range is established all along the capillary, and the different species will be migrate until they reach the pH corresponding to their PI, isoelectric point. Sorry, and at that time, when they are when the pH is exactly the same as the isoelectric point, their global charge is near zero, and they stop to migrate. Okay, so that's why how the different charge variants are separated, because each charge variant, as you can see here, has exhibit different PI. Okay, So you obtain a profile, and you can see here the different type of profile obtained. So here is it's the main peak, and as exactly as described with IEX, you have on the right the basic variant, on the left the acidic variant. And like this, you obtain a profile that could be used for identification. And so here is the two maps have been, and which is and why I said it's a good for, sorry, for identification. I forgot. Here is a map. It's the map. It's a reference map of reference, and here is infliximab, which is a therapeutic map used against the Crohn's disease, and is able to recognize the TNF alpha. And you can see here the profile obtained for infliximab. So you can see when you compare these two maps that the profile is different. So which rendered this method really interesting for identification, identity purpose, identity issue. And then I think that's it. All I want to present. I think or maybe it was I can. So that's it for all the method, the separative method that can be used to for quality control issue in terms of identification, in terms of detection of degraded species, truncated or aggregate. Et alors après, and then that's it for today. J'ai fini un peu plus tôt que prévu, c'est pas grave. Okay. Is it okay for you? Okay. Hay más acá.
¿Tiempo para preguntas?
Yo voy a hacer una pregunta, pero en inglés.
Tito, traduis après.
Entiendo en inglés.
I have. Well, I finished with the antibody, but in fact, I have with advanced therapy different. But I will maybe tomorrow.
So basically, when a company is designing a new antibody, they will try to see all the potential degradation. Then they will establish like an identity map of the antibody and the potential non-good antibodies or degraded or truncated antibodies with a different technique, and then they will provide to the hospital with the quality control method to check.
No, usually they do. All the quality control have been established before their release for the hospital. But then the thing is that we know that the molecules are complex, and the hospital have not really a proper method quality control for themselves. But usually, but it happens that the maps has been released, was commercialized, but then withdrawn because of unwanted problem. It happened for atezolizumab, and mainly because he was non-glycosylated, which and that give that induce alteration of it was against the PDL2, if I remember, and it's this general alteration of his conformation and really strong immunogenicity problem.
So the hospital has to set up its own method.
Yes, but which not. But the moment it's really there is few methods to set up. There's few methods existing at the hospital. They have not set up so much. They have in France. I know they have one in Georges Pompidou, but it was Emmanuel Jacoulet who did that. But it's their own method, and they have no. The company did not give them this control has to be made. They have no method from the company.
It's surprising because they already evaluated all the possibilities.
Yes, but sometimes it's happened that a drug product is released and then withdrawn of the market because of unwanted drawback that has not been established or detected before. It's happened.
T'as compris, Ricardo?
Oui, j'ai compris.
Yeah, a bit of a related question. I was wondering if you can, when we speak about quality control, something that is very important is like the regulatory authorities and regulations. And so I was wondering also for the methods, but when you define the critical quality attributes, is that something that you as a developer or the industry establishes, or is it the regulatory authorities, or they have like
It's both. It's both. It's established with the experience. It's both. People observe, and then it's the regulator, and in agreement with the regulatory authority, this is set up. But the own company has also a reflection about that themselves. And normally, and sometimes to establish this type of quality control attribute, they also discuss with experts of this field, who could be private or academic. But it's all the time, it's evolving all the time, because new one could be considered, which was not the case at the beginning, but it appears afterwards. It came out, came up afterwards.
Les techniques chromatographiques traditionnelles sont toujours valables pour tout ça?
Oui, elles sont toujours très utilisées. Parce qu'en fait, ce que je n'ai pas montré là, maintenant, tu utilises des nouvelles colonnes, tu fais de l'UPLC avec plus haute pression, ça va plus vite qu'avant.
Ouais.
Donc, et puis bon, il y a aussi tout ce qui est la détection, miniaturisation, puis aussi la masse qui va avec. Donc les méthodes sont quand même bien améliorées par rapport à avant. Simplement, tous les hôpitaux ne sont pas équipés avec des masses. Ouais. Ça reste quand même la méthode de référence pour les hôpitaux, ça reste la chromato, ouais. En fait. Donc ça, oui, ça reste toujours. Hein?
Ça, tu vas en parler demain.
De quoi?
De ce que tu viens de dire.
Je peux. Tu voudrais que j'en parle de?
Non, je pose la question.
Non, je savais pas. Mais bon, je peux en parler si tu veux. Mais en fait, oui, je voulais parler de. J'ai pas tout fini, mais enfin, après, je savais pas. Parce que je voulais aussi parler des biologiques pour les advanced therapies, ce qui commence. Parce que par exemple, in the field of advanced therapy, they are still ongoing. In fact, they are not settled completely.
Yo había preguntado si las técnicas cromatográficas convencionales todavía son bien robustas para desarrollar esto. Me dice que sí, porque los hospitales finalmente no tienen los medios, ni el espacio, ni los recursos para implementar técnicas más sofisticadas como la cromatografía de alta presión, la UHPLC, o masa incluso. ¿Más preguntas de ustedes? Pueden hacerlas en español y traducciones.
¿Qué tan precisa es la cromatografía por exclusión, la SEC? What is the capacity of the sensitivity of that chromatography?
Sensitivity in terms of concentration or sensitivity?
In weight.
In weight. It depends of a lot of things. It depends of the type of colon. It depends of the product you are analyzing. It depends on the mobile phase. It depends on a lot of things. It's true it's not to the separation of the species closely related in terms of weight. It's not completely ideal. And it's also difficult because proteins, for instance, I said weight, but the separation is the size, in fact. It's the size. It's not exactly the weight. And sometimes for a protein, protein could have the same weight, but they will be glycosylated in a different way. So the size are close. In this case, they will not be really well separated. So, and even for the certain protein, you have the monomers, the dimers, and it's not completely, and sometimes it's not difficult to completely separate them. Because you have also, depending of the colon and the proteins and so on, they tend to interact with the stationary phases. So you have what we call pigtailing phenomenon, and there it's which reduce the resolution capacity of the colon. The separation is not completely accurate. So the mean to improve that is to modify the composition of the mobile phase, sometimes by increasing the salt, you decrease the unwanted interaction of the analyte on the stationary phase. So, but in fact, and we tried as well to separate completely in the lab the main peak from the aggregate, and we never succeed completely. The only way we want to do it because we want to use method of detection that I haven't shown here, but we have method of detection who can, when they are coupled to SEC, for instance, they can precisely determine the molecular weight of the species analyzed, and which sometimes is important because you can have a species with monomers, dimers, trimers, and so on, and we suppose that this peak here is dimer, this one is trimer, but it's not completely sure. We cannot say, okay, it's really that. We suppose it's this, but we are not sure. And for this accurate separation, better is what we call the A4F, asymmetric flow fractionation, where you combine two forces, a longitudinal one and a vertical one, and to completely separate the different species. And that is very, very accurate to separate monomers from dimers and trimers. And when it's coupled to mals, you will have a precise determination of their molecular weight. So that's the perfect one, is this one, if you want to separate different weights, different monomers from dimers and trimers. And there is a new one where they add flow fractionation with electrophoretic force. That's for the direct, for the head of unit. C'est pour le directeur d'unité, j'en profite.
It's only 1 million euros. So it's okay.
Yes, no, that's no. No, we cannot buy that alone. But
¿Más preguntas? Plutôt général, mais par rapport à ce que tu viens de montrer et les autres confs que tu nous as donné des cours.
Ouais.
Ici au Chili, l'autorité sanitaire,
oui.
pour accéder à une thérapie avec des antibodies.
Oui, des anticorps, oui.
Ouais. Il y a un département qui fait l'analyse.
Oui.
Je mélange anglais.
Oui, c'est pas grave. Oui.
Et qui s'appelle Évaluation des technologies sanitaires. Donc,
oui.
Le problème, c'est les prix des technologies, des nouvelles technologies. Comment ça se passe en France?
Alors,
l'industrie.
Ah oui, c'est cher.
Et après, il y a quelqu'un qui doit autoriser tout ça.
Oui. Bon, ça, alors, je ne suis pas spécialiste là. Ouais. Ce que je sais, c'est que c'est considéré comme un produit. Alors, c'est un produit issu des biotechnologies. Ouais. C'est pas une advanced therapy. Donc, elle a. I speak in English. They follow the rule. Oh, il traduira? Oui.
Mais tu n'as pas traduit ta question.
Oui, tu n'as pas traduit ta question en espagnol.
Ah, oui. No, pero entendí. No, les decía que en Chile, cuando hay una nueva tecnología que está desarrollando anticuerpos, ¿cierto? O fuera y se quiere importar, el ISP, el Instituto de Salud Pública, tiene un departamento, o el Ministerio, Departamento de Evaluación de Tecnologías Sanitarias, ¿ya? Entonces muchas veces se evalúan, pero considerando el precio no se incorporan, por ejemplo, en las canastas del GES, ¿ya? Entonces le pregunto cómo ocurre eso en Francia, considerando que es una pregunta más bien general que específica, pero que toca lo que tiene que ver con control de calidad y también con lo que nos presentó de terapia higiénica, etcétera, en las presentaciones previas. C'était un peu long.
Oui, ouais, en fait, comme ça m'a donné le temps de réfléchir à la réponse. Ouais. Très bien. In France, there is so it's a project, and he needs to be approved. The authority who approved the commercialization of the product called ANSM, so National Agency for Health and Medicament and Drug, but I translate. Okay. So first they approve, and then we have another organization called the Commission of Transparency, who will fix who will decide of the price of the drugs, and the price of the drug and the level of reimbursement by the Social Security. So two things, which is quite important. And in fact, as the social security is the government, is the state, so of course this national agency wants the price to be as low as possible, which is not the interest of the private company. But so they have to compromise between these two things: the economic situation of France and the social security, and the interest of the company. One way now so usually at the beginning with the first MABs, the price was relatively high. But now some companies have developed what we call the biosimilar, which is the MABs, but not the MAB, not the other company. So that's decreased the price. So now it's more easier for all the MABs which has a biosimilar. But it's a compromise. But for the moment, it's the state who paid for the patient, and the price is a discussion between the authority and the company. But sometimes it's happened now that some drugs are not commercialized in France because the authority decreased the price too much, and the company did not want that. So it's a withdrawal from France, for instance. It's happened. Tu veux dire quelque chose, Nicolas?
No, it's like a negotiation with the state and the company. And recently, Trump puts a lot of pressure on Europe because the price of drugs in Europe is rather low compared to the United States, and it's pushing Europe just to increase the prices for the American company to get more money. So it's like,
But you have some, in the new therapy on gene therapy, there was one who worked very well for one type of disease, but it was $1 million the price. And it has been withdrawn because there is not enough patients and to pay it for it. They can't afford. So the development is it's a high cost, but nobody can afford. So it has been retried, which is a pity. But
More questions? With English. Go ahead.
I have another question that is also kind of general, but it occurred to me. So with biologics, it's learned that they are going to have impurities, and you're never going to have like so as a developer also, when to put a stop, like when you say, okay, this is pure enough, and the impurities, or or it's not heterogeneous, but I do not have to kill myself in making this pure because
but
it's okay.
That's the role of they call the quality control attribute, to fix a range that has not to be exceeded. And it seems that it's work because you have sometimes unwanted response with MABs, but they also save lives. I mean, the treatment works. So you have, but it's true that you have person who has immune response unwanted, but nobody knows exactly what is the problem. That could be just the drug itself, the product itself, would generate maybe because of the small aggregate, because there is maybe a host cell protein who is stick, because another thing that nobody knows, you generate unwanted response. Nobody never never establish a link between the percentage decided by for the percentage established for quality control attribute and the unwanted or drawbacks of the drugs. In my knowledge, it has never been established.
Which technique is better, electrophoresis or HPLC?
It depends. So you ask me to choose between the team of Pierre Chaminade and my team. That's right. No, I joke. Not okay. No, in fact, I do both. I did both. For me, capillary electrophoresis is very nice as well because the The technique, you can do a lot of development, you can play with a lot of parameters, you have a lot of things has to be done. But it's true that for me, chromatography is nice as well because it's really established, it's robust, it's more easier. Because sometimes when you encounter a problem with capillary electrophoresis, you know when it starts, but you never know when it's going to be, when it's ended. So it's more tricky. But it's nice because you have many things to do because it's small, because you can automate it very easily. You can combine many things, many mechanisms. So it's very nice, and you can do a lot of things. But if you want to set up something robust that you don't want to be bothered every day, I joke, but often maybe chromatography is nice too. But that's my personal opinion. So maybe. Other colleague could have another opinion.
Do you need any additional assay to demonstrate biosimilarity, like X-ray crystallography or another?
We can need. It's true. I didn't present, but it's true that when a protein starts to be to exhibit a conformal modification, you need extra techniques. Here you can see that, and even me in my research, we have submitted maps to stress. We didn't see anything by any small aggregate by size exclusion chromatography, but by circular dichroism or even by HPLC coupled to ion mobility, we see conformational modification. So of course, separative method will not give you all the information needed. I agree with you. But it's the most used at the beginning when you start to check. If you want to check rapidly something, it's the first one used. But I agree with you. If you want to go deeper, you need orthogonal methods.
J'ai une question d'une collègue qui travaille à Santiago qui est connectée à la réseau. Nicolas, tu pourras traduire? Can you translate?
En espagnol.
Para cubrir el costo de cada medicamento, hay un seguimiento de los pacientes establecido de parte del Estado francés.
Pour couvrir le coût de chaque patient, il y a...
Ouais, mais à l'anglais. Sinon,
eso no puedo hacer.
J'ai choisi.
Tu peux redire en espagnol sur les docteurs.
Dice, para cubrir el costo de cada medicamento, ¿existe un seguimiento de los pacientes?
Les coûts.
Suivi.
I have no idea about that.
En el fondo, porque pregunta es del Instituto de Salud Pública.
Pero no somos expertos.
No.
En el fondo, por si se presentó algún efecto adverso, a eso se refiere, al aplicar ese medicamento a un paciente, el Estado verifica eso al momento de aprobar.
I don't think so. I think the pharmacovigilance is
after the once the medication is released. Yes.
Thank you. More questions? Es el momento. ¿No? Okay. Bon, merci, Claire.
De rien.
Un aplauso. Y esta tarde entonces nos reencontramos a las 14:30 en la facultad, ¿cierto? Farmacia.
Sí,
En la
facultad de farmacia, vamos
Sí.
a seguir con microtecnología. Ahora
Microencapsulación,
aplicación oral
sí.
y celular.